All right, good morning, everyone. Let's get going here. Okay, so just a quick reminder before we get into our content for today. The weekly quiz, as I mentioned last time, I'll make it available immediately after class. It's gonna include today's material, Friday's material, and last Monday's material. So those 3 lecture days, 3 questions each, which will be ventilation, diffusion, and then what we covered today, which is the transport of gases, okay? And that'll be due on Wednesday before class, just so you can have an opportunity to practice. And then whatever questions you may have, you can bring them to the review on Wednesday. So whatever we finish today will be the cutoff for the exam. And on Wednesday, we will dedicate that to a full day of review. Okay, so if you have questions, you can email me before then or bring them to class on Wednesday. Now, before we move on to the next packet, I got some questions about this last table. So we talked about a few of the terms on this table. We talked about hyperapnea, hypercapnia we spoke about, and hyper and hypoventilation. The other terminology, not memorized, but be familiar with some of this terminology. The ones you should definitely know are hyper and hypoventilation and hypercapnia, but some of the other terms you may see on a question, and that can help you eliminate um, options, or just be familiar with these terminology, not necessarily memorize them or completely ignore them, okay? All right. Okay, so we're going to move on today to speak about the transport of gases in the blood. And this is really going a step further from what we talked about in terms of diffusion. So when we looked at diffusion, we were really talking about those partial pressures and how that gradient was set up to actually move the gases through the circulatory and pulmonary systems. But today we're going to speak about how these gases are loaded and unloaded to hemoglobin specifically, and then the other mechanisms for transport as well, okay? So we'll start off by recalling the fact that we said oxygen is not very soluble in the blood. This is something we spent quite a bit of time when we looked at those beakers establishing this fact, right? Oxygen does not move around very well in the blood. And so we've got this hemoglobin that is crucial to the movement of oxygen around the system. So only 1.5% of our arterial blood has oxygen dissolved in it. This is so little that if we were to rely just on this amount alone, our cardiac output would have to increase about 50 times what it currently is in order to adequately oxygenate all of our tissue. So this is very insufficient in terms of the amount of oxygen that we need. So the remainder of the oxygen to meet our demands is circulated using hemoglobin. And we talked about this reversibility of hemoglobin. Hemoglobin is that 4 subunit protein molecule that can bind oxygen onto those heme groups that we looked at previously, and it can do so reversibly. So it can bind oxygen tight enough to move it around the system, but not too tightly that it cannot be dissociated when it gets to the tissues to oxygenate those tissues. So we've got this reaction, and we've got these double-headed arrows that indicates the bidirectionality here. On the left, we've got deoxyhemoglobin, which is the hemoglobin not bound to oxygen. And on the right, we've got oxyhemoglobin, which is the form when it has those 4 hemoglobin molecules bound. Now, what we can see in this image is the partial pressure of oxygen across the alveoli, and this is facilitating the diffusion of oxygen from alveoli into the pulmonary capillaries. And if you look more closely at what's happening here, this is the alveoli, this is oxygen, it's moving across this membrane, which is the alveolar cell, the fused basement membrane, and the capillary cell. What do we call that membrane? Just for review. What do we call those 3 structures? The respiratory membrane, I'm hearing it right. So the respiratory membrane. So oxygen is moving across the respiratory membrane here. It's gonna move into this erythrocyte. It's gonna bind on to hemoglobin, and it's gonna circulate through our systemic arteries, eventually capillaries, and then eventually at the tissues. If we look more closely at what's happening at the tissue level, we can see the opposite happening, right? Oxygen dissociating off of hemoglobin, crossing the capillary bed, and moving into the tissues where it's being consumed for cellular respiration. So it's really important that hemoglobin bind oxygen reversibly, and we're going to move on today to speak about what influences the different states of hemoglobin. So what tells hemoglobin to want to bind oxygen, and then what tells hemoglobin to want to let it go when we get to the level of the tissues. Okay, now we know hemoglobin can bind 4 oxygen molecules. It's got 4 heme groups in the center of those subunits, and we can bind an oxygen each. And so when we look at the law of mass action, this law states that if we've got more of our reactants, we'll drive the reaction towards products. And if we've got more products, we'll drive the reaction towards reactants. Okay, so if you've got more oxygen around, we're gonna bind more oxygen to hemoglobin. We're gonna create more oxyhemoglobin. And if you've got more oxyhemoglobin, we'll drive it in the opposite direction to create more deoxyhemoglobin. And so at complete saturation, hemoglobin has all 4 of its subunits, so all 4 of its heme groups in those subunits bound with oxygen. And this can go back and forth depending on a few factors that we'll speak about. Okay, so the saturation of hemoglobin is really an important factor that we're gonna determine. So this is it in its 100% saturated state. Okay, everyone good here? Any questions? All right. So let's look at what's happening in terms of hemoglobin and it becoming saturated versus unsaturated. So we know that when we have 100% saturation, hemoglobin has the ability to carry 1.34 milliliters of oxygen. And typically in an adult human without any sort of pathology or anemia, we have anywhere from 2 to 17 grams per deciliter of hemoglobin. So if you multiply those factors, the oxygen carrying capacity of hemoglobin is about 200 milliliters of oxygen per liter of blood. That's how much oxygen we have circulating in a liter of blood. And we can even go a step further and talk about our cardiac output, right? We know cardiac output to be what? Okay, stroke volume times heart rate, but in terms of a quantity, how much, does anyone remember? It's kind of a stretch. 5 liters, right? So we got 5 liters per minute circulating, and we've got 200 milliliters of oxygen per liter. So we've got 1,000 milliliters of oxygen circulating our body per minute, okay? Now, if we look at the different states of hemoglobin, when it is 100% saturated, or actually it's 98% saturated because all of the hemoglobin aren't actually bound in actuality, but we can think of it as being 100%, as being full. This is in our arterial blood. What we see is that each of those subunits, each of those heme groups has an oxygen bound to it. We can see that here, with the exception of a few. That's why this is not fully 100, but for our purposes, we'll consider it to be 100, okay? And so at a partial pressure of 100, which we talked about, that's the partial pressure of oxygen in our systemic arteries. Hemoglobin has most of the subunits bound with 4 oxygen molecules. Now, when we look at our venous blood, when hemoglobin is about 75% saturated, we don't actually lose all of the oxygen, right? We only lose 1. So in the unsaturated form, hemoglobin is only releases or only loses 25% of its oxygen carrying capacity. That's a really, really important phenomenon. So hemoglobin is never empty, all right? It's never completely lost all of the oxygen molecules bound. Can anyone think of a reason why that would be important? Can we think of a reason why that would be important? So even in deoxygenated blood, even in our systemic veins, we've still got oxygen bound to hemoglobin. So yep, that's part of the answer. So it's going to help to drive the reaction in the opposite way. It's going to help with the state of hemoglobin to move towards going back to the lungs. But something else in terms of protecting our tissues. So this is actually another one of those protective mechanisms, okay? So we can increase our metabolic demands, right? Tissues can increase their metabolic demands up to 4 times, and we still have adequate oxygen to supply. So at rest, our tissues are only requiring 25% of the oxygen that's being circulated. And so in instances of an increased metabolic activity, exercise, a fight or flight response, we have lots of oxygen circulating as a backup reservoir for increased metabolic activity, okay? All right. So this brings us to the oxygen hemoglobin or hemoglobin oxygen dissociation curve. So this is how hemoglobin changes its shape, changes the way it behaves, depending on the partial pressure of oxygen and how it loads and unloads those 4 oxygen molecules. So we'll describe this here a little bit and then we'll illustrate this. We're going to do quite a bit of drawing because we're going to draw out some different scenarios here. But let's go through this here to start. So on the y-axis, we've got the percent of oxygen saturation of hemoglobin. And that's a really complex, fancy way to say how many oxygens are bound on that hemoglobin, whether it's 1, 2, 3, or 4. Okay. On the x-axis, we've got the partial pressure of oxygen. Again, another fancy way of saying how much oxygen is present, right? Depending on where we are in veins, we've got less. In arteries, we've got more. And then if we look at the way that this curve behaves, it's not a direct or a linear relationship. It's actually more of the sigmoidal curve. And that's because when we bind 1 oxygen, right, so about 25%, right, because we know 1 out of that 4 is 25% saturation. If we bind 1 oxygen here, we automatically have this conformational change in hemoglobin such that it increases its attraction for oxygen. And so the other 3 oxygens are going to bind with much more ease because of that change in conformation. So we see this more steep curve as we bind those 2nd and 3rd oxygens on. And then when we get to the top here, as we've bound that 3rd oxygen, we see the graph flattening off because we now have less spaces to bind, right? We've bound that 3rd oxygen on. And so the graph flattens off as we get to that 4th oxygen. And as we talked about on the previous slide, we really only change the this final quarter, this final state of hemoglobin is what we change because we're really only using this 25% of the oxygen that's bound here. So in healthy tissues, we're going to unload just that 1 oxygen and we're going to keep hemoglobin circulating around. Okay. Any questions on that bit so far? Any questions so far? Okay, so what we'll move on to see is that this curve can shift to the right or to the left based upon a few different circumstances. Okay, so we're looking at the hemoglobin, and I abbreviate this with HB, oxygen dissociation curve. Okay, we're going to draw a similar graph here. We've got the partial pressure of oxygen on the left. That's in millimeters of mercury. On the x-axis, did I say on the left? I meant on the x. On the y-axis, sorry, we've got the percent hemoglobin, oxygen saturation. And all that means is how many of those 4 oxygens are bound to hemoglobin. Right, so we've got 100%, got 75, 50, and then 25%. Okay, so we got 1, 2, 3, or 4 oxygens bound. And we talked about the shape of this graph being this sigmoid curve. Right, the sigmoidal curve. And that is really describing what we call cooperative binding. And cooperative binding means that oxygen binds more easily to hemoglobin after that first oxygen has bound. So after that first oxygen binds, we then have that steep rise in the curve, and then it flattens off. Okay? Now, we know that, so this is our first oxygen. We know that when we get to 75% oxygen saturation, this is the level of the partial pressure in our systemic veins, which is about 40 in terms of what we talked about last time, right? So at a partial pressure of 40 millimeters of mercury, we have bound 3 of our 4 oxygen molecules. Okay? And so this is a standard curve. So this is the way oxygen behaves, or hemoglobin behaves in terms of oxygen binding under normal conditions. What we're going to speak about now is when the curve shifts to the right and when the curve shifts to the left, and what are some factors that causes that. Okay, so let's describe the right shift first. So when the curve shifts to the right, hemoglobin has a lower affinity for oxygen, okay? It doesn't want to bind oxygen. It's going to unload oxygen easier, and it's going to load oxygen harder. So we've got a lower affinity for oxygen, okay? Meaning that it's going to be harder to load oxygen and easier to unload oxygen. Okay? And we're going to talk about a lot of different states that align with this right shift, but we'll mention 2 things here now. This happens under the instance of increased body temperature, as well as increase in altitude. Okay, so increased body temperature, increase in altitude. And really, anytime we have increased metabolic activity, we have a right shift of this curve. Okay, so increased metabolic activity, and we can think about exercise as a really good example in that. Now, when the curve shifts to the left, we've got the opposite scenario here. So a left shift we have a higher affinity for oxygen, meaning that it is harder to unload oxygen, and it's going to be easier to load oxygen because of that increased affinity. This happens when we have decreased body temperature or when we have a decrease in altitude. Okay. All right, so I really want to explain why we have this right shift in terms of the big picture of what's happening. So the tissues are requiring more oxygen if we shift to the right. We're thinking about things like exercise, we're thinking about things like increased body temperature. We're going to talk about some other factors as we go on that shift the curve to the right. But if you kind of associate all of this with increased metabolic activity, then the other factors that we'll speak about will make a lot more sense, okay? And so what's really happening with this right shift is that we need a higher partial pressure. So a higher partial pressure of oxygen is required in order to fully saturate hemoglobin. So a higher partial pressure is required to saturate hemoglobin. Okay, so it's harder to get that oxygen to stay on hemoglobin. They don't have as much of an attraction or affiliation for each other, and that's kind of what we want in these types of scenarios. We want that oxygen to dissociate easily because these are instances when the tissue really needs oxygen, right? Increased metabolic activity, increased body temperature, even increased altitude. We know that at increased altitudes, we have what in terms of a partial pressure? Higher or lower? Lower, right? The higher we go, think about the top of Mount Everest, we've got a very low partial pressure. So we've got less available oxygen. So we don't want hemoglobin really affiliated or really binding tightly to oxygen. We want it to dissociate off easily. And so all of these instances align with that easy dissociation. At a left shift, this is the opposite, right? We can here afford to have oxygen binding tightly to hemoglobin because we just don't need it that much in the tissues. And so we need a lower partial pressure required to saturate hemoglobin here. Okay? All right, I hope that makes sense. And we're going to go on to speak about some other scenarios that shift this curve to the left and to the right. All right. Any questions? Any questions? Okay. So let's jump back here. We're going to do quite a bit of drawing today. I apologize in advance. Okay. So the second factor that influences the way that hemoglobin behaves is called the Bohr effect. So this is the influence of pH on the behavior of hemoglobin. And what we see is that when we have an increase in pH or a lower pH rather, and an increase in acid, we see that oxygen unloads more easily, okay? What are some instances where we see increased acid? Lactic acid, for example. Increase in exercise, right? Which is another thing that increases metabolic activity. So increase in acid or lower pH increases the unloading of oxygen and shifts our curve to the right and makes oxygen and hemoglobin really unattracted to each other so that that oxygen can dissociate very easily. That's called the Bohr effect. So if we produce more acid or decrease our pH in tissues, we shift this curve to the right, and this allows oxygen to unload more easily. Alrighty? Okay, so let's illustrate that here again. Okay, so we're describing this as the Bohr effect. And we talked about this equation, right? So hemoglobin, deoxyhemoglobin plus oxygen can become oxyhemoglobin. And then we've also got some protons released. Okay, let's just label this out. Now, given the law of mass action that we spoke about, so the law of mass action dictated to us that if we have more products, we drive the reaction towards reactants, and if we've got more reactants, we drive the reaction towards products. So if we have more acid, which is one of our products, and remember, more acid is lower pH. So high acid, low pH, low acid, high pH, okay? So this is a low pH. Then this drives the reaction to the left, right? And another way that we can think about this is that if we've got more acid or more protons, then oxygen is going to dissociate from hemoglobin more readily, right? As we drive this reaction to the left, oxygen kicks off of that hemoglobin and we end up with deoxyhemoglobin, right? So oxygen dissociates easily, okay? And what's another way we can think about that in terms of our shift, right or left? Based upon what we just drew on our previous slide. To the right, right? So if oxygen doesn't want to be on hemoglobin, that's shifting our curve to the right, okay? So another way we can think about that is a right shift, right? So let's draw this out here, kind of make more sense. So we've got our partial pressure of oxygen. We've got our percent hemoglobin, oxygen saturation. Okay. We've got our standard sigmoid-shaped curve. Then we've got a shift to the left and a shift to the right. Okay, so what we're saying here is that at decreased pH or more acid, remember protons are acidic, so this is what I'm calling acid, these protons. This causes a right shift. And if we're thinking about the overall picture, like I mentioned earlier, this is again, increase in metabolic activity. So think about things like lactic acid production, right? Lactic acid, which is going to drive this curve to the right by, which is going to shift the curve to the right by driving this reaction to the left, okay? Think about lactic acid production, which is an increase in metabolic activity. Okay, so if you're really understanding why the curve shifts to the right, you really won't have to memorize each of these points so much as understanding why acid does what it does because of the bigger picture of that metabolic activity increase. So when you shift the curve to the left, this is caused by an increase in pH. So if the pH becomes more alkaline, right, or a decrease in the acid production, and that causes a left shift. Okay. All right. Any questions on this part? Any questions? Okay, we're only going to draw this one more time. So that was the influence of pH. Okay. The third factor here is carbon dioxide itself. Okay, so carbon dioxide exhibits what's called the carb-amino effect, and that is the way carbon dioxide influences, again, this shift in the way that hemoglobin behaves because of the presence or absence of excess carbon dioxide or too little carbon dioxide. So CO2 reacts with hemoglobin to form what's called carb-amino hemoglobin. And so if we have hemoglobin plus CO2 and they bind, we talked about the fact that hemoglobin does have the ability to move some carbon dioxide around. So there are three mechanisms for transporting carbon dioxide. We're going to speak about the other two, but carbon dioxide can be mobilized on hemoglobin to a lesser extent. And so when we have this carb-amino hemoglobin, this lowers the affinity for oxygen. Okay, so basically the big picture here is that in excess states, or if there's excess carbon dioxide, hemoglobin does not want to bind onto oxygen. It wants to let it go or to dissociate it. Okay, so this again is aligning with that increase in metabolic activity. Remember, CO2 is a byproduct of respiring tissues. And so the more we have metabolic activity, the more CO2 will produce as a byproduct. And that'll shift our curve left or right. Right. That'll shift our curve to the right? So again, aligning with that increased metabolic activity created by having more CO2. And if you think about the underlying physiology here, if tissues, again, are respiring more, are absorbing more CO2, are absorbing more oxygen, producing more CO2, then we don't want oxygen to be very tightly bound to hemoglobin. We want it to be loosely bound so that it can dissociate off very easily to match the demand of those respiring tissues, okay? Okay. So here we're looking at the carb-amino effect. Is that a question or stretching? Okay, it's okay. Any questions at this point? I hope I'm not going too fast. Okay, so the carb-amino effect is where we looked at the fact that hemoglobin had this ability to bind CO2. And again, this is a reversible reaction, so we can bind it on. This gives us carb hemoglobin. Right, we call this carb-amino hemoglobin. When it has that CO2 bound on. And again, we're looking here at the law of mass action, just to make sense of this, which states that if we have increased CO2, which is our CO2, which is our product here, that drives the reaction to the left. Okay, and again, if we are driving the reaction to the left, we're simply dissociating oxygen off from hemoglobin. Okay. Which is right shifting. Okay, so this is also aligning with that increase in metabolic activity. And we'll just illustrate that here. Although this should be very Okay, this should be very familiar at this point. This should make sense, right? So we've got our normal curve. We've got our right shift, and we've got our left shift. So shifting to the right is caused by an increase in CO2. And we know that increase in CO2 aligns with increased metabolic activity. Okay, and then decrease in CO2 causes a left shift. Okay. So far we've talked about quite a few factors that influence the way hemoglobin behaves. We're going to talk about one more factor, but so far we talked about temperature. So increased body temperature, we talked about partial pressure, right? Increased partial pressure. We talked about altitude, higher altitudes. We talked about carbon dioxide, increased carbon dioxide. We talked about pH, which is lower pH or increased acid. Those factors shift our curve to the right. And all of those factors align with increase in metabolic activity, okay? And then factors that shift to the left were decrease in temperature, and then we talked about lower altitudes, sea level and below sea level, okay? All right, so okay, what questions do we have before I move on? What questions do we have? Okay, now the fourth factor that influences the way hemoglobin behaves is called 2,3-DPG, aka 2,3-BPG. So you may see this in different literature in different ways, but just understand it's the same thing. 2,3-diphosphoglycerate or biphosphoglycerate, referring to the same molecule. So this is produced in red blood cells under conditions of anemia or low oxygen saturation. This is also produced at high altitudes, which also equals low oxygen saturation. And so basically under these types of states, we create more of this 2,3-DPG. And the way that 2,3-DPG acts is by blocking the interaction of hemoglobin and oxygen. Okay, so it's going to basically kind of block hemoglobin from wanting to attract or to bind oxygen. And it's synthesized in response to anemia. So that makes sense, right? If you think about that. Under instances or situations of anemia or low oxygen, I don't want hemoglobin to be bound very tightly to oxygen. Again, I want it to dissociate off very easily. I want it to enhance the way that oxygen unloads from hemoglobin. And this is why 2,3-DPG is created under instances of anemia or high altitude where we have low partial pressures of oxygen, okay? And then we also want to talk about, so let's look at this curve here. So we're seeing oxygen saturation in the absence of 2,3-DPG and oxygen saturation with 2,3-BPG present. And all we're seeing here is a shift to the right, meaning that we need a higher partial pressure of oxygen in order to fully saturate hemoglobin, because hemoglobin doesn't want to bind easily to, doesn't want to bind that oxygen easily, okay? At the bottom here, we're going to mention carbon monoxide. Again, we talked about carbon monoxide a little bit in chapter 15, and it's basically this alternate molecule that has an ability to bind onto hemoglobin, and because it does so irreversibly, and it also does so at higher affinities, meaning that it's more attracted to hemoglobin than oxygen is, what we see is that it prevents oxygen from binding onto those heme spots. So it competes with oxygen molecules for those heme spots. And as we can see, this can become fatal, right? If we can't bind oxygen because carbon monoxide is kind of in the way, then we cannot circulate oxygen, we cannot oxygenate tissues. Okay, now we'll speak a little bit about the role of carbon dioxide, the way carbon dioxide is transported. And this brings us to speak about carbonic anhydrase. And this is something we also introduced in chapter 15. So carbonic anhydrase catalyzes the reaction of carbon dioxide plus water to form carbonic acid. And that is really how we circulate the majority of our carbon dioxide. So carbon dioxide exchange we know happens in the systemic tissues. What direction is it moving in systemic tissues at the capillary beds? So is it going from tissue to capillary or capillary to tissue? Tissue to capillary, right? And in the pulmonary circulation, it's going from capillary to alveoli, right, to be exhaled. And the way that oxygen influences this is that in the presence of CO2, oxygen again shifts to the right and unloads very easily from carbon monoxide, from hemoglobin. Okay, so in this table, we're comparing the three mechanisms for circulating carbon dioxide. It can be dissolved in the blood to a lesser extent. It can be circulated as bicarbonate, and this is the reaction that is catalyzed by carbonic anhydrase, or it can be bound to hemoglobin, as we saw, again, to a lesser extent. And so what I really want to point out here is that there is more CO2 dissolved in our blood. Remember, we talked about only 1.5% of oxygen being circulated in the blood. Here we've got 5.5% of CO2 circulated. But a lot of, the majority rather, of that CO2 is bound, is circulating via that carbonic anhydrase mechanism. So this is the majority of our CO2 circulating through that carbonic anhydrase mechanism. And to a lesser extent, we can have it freely dissolved or we can have it bound to hemoglobin. And so this is that reaction. So we've got CO2 plus water catalyzed by carbonic anhydrase to give us carbonic acid. So this is carbonic acid. And that can freely dissociate to protons and bicarbonate, okay? And those protons and bicarbonate is what moves around in the blood, and then they are catalyzed in the reverse direction when they need to be exhaled at the level of the lungs. And so again, if we're looking at the law of mass action here, increase in CO2 levels drive this reaction towards our products, bicarb and protons, hydrogen ions, and then increase in protons or hydrogen ions and bicarb drives the reaction in the reverse direction towards the production of CO2 in the lungs so that CO2 can be expired. Okay, so really important to understand here that depending on where we are in the body, what tissues we're at, this reaction is being driven in the right or driven to the left in order to make bicarb and hydrogen ions or to make CO2 and water to be exhaled in the lungs. Okay. All right. Now this graph is comparing the total amount of CO2 against the partial pressure of CO2 and the way it behaves differently for different amounts of oxygen. So at a oxygen saturation of 0, right, we've got no oxygen in the blood, to a partial pressure of 100, right, which is what our systemic arteries look like, we can see that the amount of CO2 in the blood differs, okay, for the same partial pressure. So if we have no oxygen present, then we've got lots of CO2 circulating in the blood. And if we've got lots of oxygen present, then we've got little CO2 circulating in the blood. So this is really showing us that oxygen influences how much CO2 is circulating or mobilizing in the blood. And this is looking at all three of those mechanisms we talked about, right? Bound to hemoglobin, freely dissolved, and using that carbonic anhydrase mechanism. So using all three of those mechanisms, we can transport less CO2 if we've got more oxygen, and we transport more CO2 if we've got less oxygen, okay? All right, any questions on that bit? I won't go into we'll go through the flow charts at the beginning of our review session, just because it's going to get a little bit more complex. Any questions at this point? Okay, I'll see you guys on Wednesday.